All right, welcome everyone. Today we are starting 1 minute earlier than the last time, so hopefully by the next few lectures we'll actually start on time. I apologize for the delay. Today is the start of the actual foundational material of the course. So today you're actually going to learn something. Last time it was just a preview. And what we're going to learn about today is algorithmically foundational. So for almost any problem in computer science and for almost any problem in machine learning, you will need to somehow create optimal solutions. And what we're going to learn about today is a foundational technique known as dynamic programming for searching through a space of exponentially many possibilities to find in polynomial time the theoretically optimal solution. Basically, you're guaranteed to find the optimal solution, even though there's an exponential number of solutions. So it sounds like magic, but it's really coming down to smart algorithmic thinking, ordering the computation in a way that enables you to have results available when you need them, and then computing the answer to larger problems as a function of the previously computed answers to smaller problems. And the specific topic that we're going to be applying this foundational technique to is sequence alignment. Basically, how do we take two sequences and align them to each other? So as promised, this lecture has many dualities. The first is biology and computation, and then we're going to see some more things that emerge. But basically, the foundational biological problem is by itself one of the most important problems in biology, namely, how do we infer the evolutionary events that relate two sequences from two different species to each other, or two genes from the same species to each other through ancient duplication event. So sequence alignment and dynamic programming, for lack of a more creative name. So here we are, beginning of module 2, aligning and modeling genomes. So in this module, we're going to learn about dynamic programming. We're also going to learn about hashing. So on Thursday, we're going to learn about hashing, another foundational technique pervasive in computer science. And then we're going to learn a very important, commonly used machine learning technique known as hidden Markov models that actually build on the same principles of dynamic programming that we're going to learn about today. So basically on the computational foundations, we're going to learn about how to explore exponential spaces in polynomial time. We're going to introduce hidden Markov models, and we're going to look at multiple hidden Markov model algorithms. And then we're going to learn through that foundational machine learning techniques, like learning the parameters of a model, applying the model to the data, seeing if a model is a good fit to the data. In the context of HMM, we're going to call them decoding, evaluation, parsing, calculating the likelihood, and then scoring the model. So this week, sequence alignment and comparative genomics. And next week, how do we model genomes? How do we learn about exon, intron structures, CPG islands, and so on and so forth? Okay. So as you all know, we are the descendants of the first digital computer. It kind of looked like this, only even simpler than that. It was the first living thing on the planet, and it was based on some nucleic acid. As we're going to see in a lecture later in this course, most likely an RNA molecule rather than a DNA molecule. RNA was invented much earlier than DNA, and RNA invented proteins, and together proteins and RNA invented DNA. Of course, not invented in our sense, but created somehow, evolved. All right. So all of the species that you're looking at today are coming from the same common ancestor. It's weird, right? Like a mosquito that bites you, whatever is growing in your hair, you don't want to know. Don't tell your mom. The table that you're writing on, all of that stuff shares DNA and is living and breathing and still growing around us. I mean, I don't know if you've tried this, but take Miracle Growth and put it on your table. It'll actually grow. Tables are still alive in many ways. So anyway, all of these organisms come from a single common ancestor. So if you look at the tree of life, it is incredibly rich. If you're kind of bored with the life forms that are above the water, put on a mask and go below the water. Your brain will explode. Hopefully not because of the pressure. Don't try this at home. it is mind-boggling to see what diversity lies in our planet, and every dive you learn about new life forms that you didn't know existed, which is quite mind-boggling. And we're kind of like the boring ones down here, but there's much cooler life forms out there. And if you look at the evolutionary inventions, a lot of them sort of are shared because they were beneficial, and then they were, you know, they led to many more species that had them so surviving in different niches. Evolution is also embracing extinctions, and extinctions are part of life. So, you know what they say about life, nobody gets out of it alive. And same for evolution. Basically, no life form that exists today has maintained itself unchanged since ancient times, and many of them have simply died off through catastrophic events, such as the meteorite that hit the Yucatan 60 million years ago here. Some of the lineages survived. I mean, we shouldn't be calling them birds. We should be calling them just this branch of dinosaurs, because they're, you know, just a tiny little branch of a much larger group of species known as dinosaurs, actually. And then if you compare these species, you can actually align them to each other. Basically, the fossil record is very rich, but much, much richer is the DNA record. And you can actually use DNA to recognize the patterns of evolution of closely related species and distantly related species. You can basically line up all the genes encoded on the plus strand in yellow and the minus strand in magenta, and then sort of create alignments that basically tell you that, wow, all these genes have maintained more or less the same order and orientation across different species. That allows you to align both coding parts and non-coding parts, and basically start recognizing functional elements across complete genomes. You can basically use that to start identifying what regions of the genome are important. So here's an alignment of dog, human, mouse, rat, and as well as chicken, fugu, and zebrafish, all on the same human reference. And you can see here the strength of conservation computed using an HMM that we're going to learn about next week, and the annotation of protein coding exons. So the tall blue parts are protein coding regions, and the tall black parts are conserved regions. So we're going to learn about how to use this to actually read evolution's notebooks, to basically figure out what regions carry out what functions, to distinguish protein coding genes, RNA genes, microRNAs, regulatory motifs, and we're going to learn all that in the evolutionary section of the course. But today we're going to focus on how do we even build these alignments. Basically, in all of these cases, I'm showing you nucleotides on top of each other. How can we build these alignments? So this was in fact the focus of my PhD thesis. So in 2003, I published this paper in Nature that basically showed how we can align multiple species to completely de novo discover all of the functional elements that we know about in those species, like protein coding genes and regulatory motifs. So you can see here an actual alignment from that paper and from my thesis, which basically looks at 4 species of yeast, Saccharomyces cerevisiae, Paradoxus, Micatai, and Bayanus, which are approximately the distances of human and mouse for the 2 furthest species, and the specific nucleotides in each of the species between the genes GAL1 and GAL10. So these genes are involved in processing galactose, the sugar found in your milk, and there are regulators that control whether yeast should be turning on the pathway to process galactose. Because if it finds sucrose, the sugar that you find in white granulated sugar, for example, it's much more effective, much more efficient. If you give my 2-year-old daughter a glass of milk or a bunch of sugar, she's going to go for the sugar. Yeast are making the same choice. So if they have bottled milk and a sugar, they're just going to go for granulated sugar. And that's what glucose is versus galactose. So there's a complex regulatory machinery that decides whether to turn on this or that gene. And that machinery is very simple. It takes in 2 inputs, whether there's glucose or galactose in the environment. If there's no galactose, none of these genes turns on. If there is galactose, GAL4, the regulator, binds a motif, which is CGG, spaced by 11 nucleotides from CCG. 11 nucleotides is basically one full turn of the double helix. So 10.5 nucleotides is how long it takes for the helix to make a full turn. So that basically means that that regulator, GAL4, is contacting the DNA on two equal parts, which are actually palindromes, reverse palindromes of each other. If you look at CGG and you read what's on the opposite strand here, CCG is actually CGG on the opposite strand. So it's actually a reverse palindrome. It's two equal arms that contact the DNA helix from the same side on two different parts. Everybody with me? So it's kind of cool, right? Like it looks just like another DNA sequence, but there's so much biology there. There's so much like information encoded within here. So GAL4 normally binds all these four sites because it really wants to activate these genes. And then it recruits these very creatively named TBP protein, TATA binding protein, which binds TATA. So there's TATA here. And then on the opposite strand here, there's TATA. Okay, so TATA binding protein binds to turn on GAL1 and to turn on GAL4. We think this is kind of cool, right? We're like reading the circuitry of a species. So all of that is about galactose. If there's galactose, those genes turn on. And what does MIG1 do? Well, if there's glucose, it binds here and it represses these genes. So it doesn't allow galactose to turn on those genes. And it instead turns off these genes because the glucose genes are going to turn on. Yeah?
So if you have like 4 galactose recognition sites, can you have just having like more lead to more upregulation?
Yeah, it's that simple. So you can actually design regulatory sequences based on these principles. If I throw more of the protein, more of the, regulatory machinery will be recruited. Of course, it depends on what is the rate limiting step. The rate limiting step might be that I only have limited TATA binding protein, or that I have only limited transcriptional machinery, etc. So, but if this is not the rate limiting step, of course, doubling the number of GAL4 binding sites means that you're going to double that amount. Who thinks biology is kind of cool? Hey, it's like principles, basic principles. All right, so why is this picture so cool? The picture is so cool because, first of all, this is the first time that this had ever been done. I mean, first, cool for me because it was the first time I was saying it. It was cool for my advisor because no one had done it before. And what I was seeing in front of my eyes were these things here that I've underlined in red, these islands of conservation. I was like, goodness, I can actually recognize conserved regions in the yeast genome. And these regions match almost perfectly known regulatory motif binding sites in the yeast genome. So that basically meant that I could read evolution to figure out what are the important parts in the known coding genome, something that had never been done before. And we could now use that across the entire genome to discover motifs de novo. And we're going to learn about that at the motif discovery lecture. We're going to basically learn how, just by enumerating k-mers of 3-gap3, I could now discover a dictionary of all regulatory motifs in the yeast genome from scratch. So computational biology is extremely powerful, but it relies on the ability to construct these alignments. And that's what we're going to learn about today. So how do we even build these alignments? How do we line up these nucleotides on top of each other? Okay, everybody sees the importance of the problem? Good. So we can read evolution to reveal functional elements. How do we actually align two genes? So I have a sequence of DNA and another sequence of DNA. I know that they're related. I know they're from the same spot in the genome. How do I put them on top of each other? So we're going to introduce, first of all, the problem of sequence alignment. Then we're going to introduce the principles of dynamic programming, which we're going to use as our solution after we realize how hard it actually is. And then we're going to combine the two and see how dynamic programming applies to sequence alignment. And then we're going to look at some really, really cool variants of dynamic programming. Okay? So, basic introduction. Genomes change over time. So if you look at, the types of events, we're now learning what are the primitives. Number one, it's mutation. So a C might, in the next generation, become a G. How does that happen? Well, that's usually that polymerase, DNA polymerase that copies DNA, makes a mistake. Everybody makes mistakes. And then another event is deletion. You might actually just simply skip. As you're copying this, you might just like forget the C and forget the A. And another one is like you might just insert an extra letter. Okay? These are primitive events, and that's pretty much all you need to explain most of evolution. So you have some beginning, you have some series of events, and then you have some endpoint. Okay? So the goal is, can we infer without the intermediate steps? How do I turn this sequence into that sequence? Okay? So we're going to look at algorithmically, how do we even define this problem? So the first is to take a biological problem and formalize it, frame it in computational terms. Define a set of evolutionary operations, so insertion, deletion, mutation. And symmetric operations allow for time reversibility. And that's very important. It's part of the design choice. Part of the reason for that is that we never have the ancestor, as I showed you, leading all the way to the modern species. All we have usually is modern species. We have human and we have mouse. And we're going to look at the series of operations that either turn human into mouse or mouse into human. But we're not going to have typically access to the common ancestor, at least not observed. Most often we're going to have some inference of it. So basically, time reversibility allows us to say, well, if there's X distance here in that direction, X and Y distance here in that direction, we're just going to put X plus Y and then think of it as if it was forward evolution. Everybody with me so far? Great. There's some exceptions, but we're not going to worry about them. So for example, CpG dinucleotides are very often deaminated, leading to a C turning into a T, and this is typically non-symmetric. The frequency of that is not symmetric. Anyway, so second is, once we have the operations, we're going to define an optimality criterion. So how do we know that we have a good solution? Maybe we're going to, I mean, we simply can't tell the exact series of operations. All we can say is maybe we're going to find the minimum number of operations that it takes. But nature, instead of just going from the minimum, most likely it took some kind of longer path. As it's exploring evolutionary space, it's making a bunch of mutations, reversing them, and so on and so forth. And especially with DNA letters, it's very easy to change an A into a T, back into an A. And then when we look at it today, we're like, oh, the A is still there. But in fact, evolution changed it twice. It's not that it's still there. It's just, you know, exploring space. So this is known as Occam's razor. So Occam was this philosopher who would basically say, among solutions that are equally good at explaining the world, I'm going to choose the simpler one, the one that has fewer parameters, if you wish. In this particular case, the one that has fewer operations. Okay, so we're going to use Occam's razor and find the minimum number of operations. But when we start going through the evolutionary section, we're going to look at the most probable or the maximum a posteriori solution. And that's going to have a lot more complexity. That's going to allow us to sometimes find that a longer path is actually more likely than the shorter path. And we're going to correct for that. So our optimality criterion in this particular case is going to be the minimum number of events or the minimum cost of events, if I can score the penalty that I get for different types of events. Who has a right arm? Who's with me? Awesome, good, excellent. Everyone has a right arm. And then once we have our primitives, the set of evolutionary operations, and our optimality criterion, we're going to now design an algorithm that either achieves this optimality or gets close to that optimality, okay? And then the tractability of the solution very much depends on the assumptions in the formulation. Why am I saying all this? Because you guys are going to be facing the same problems in your final projects. You're going to basically take some biological problem, you're going to formulate it computationally, and you're going to have to make a series of assumptions. Some assumptions are going to make the problem closer to biology. Some assumptions are going to make the problem more tractable. And the two are typically going to be in opposite directions. Sometimes they're aligned. Sometimes better modeling the biology, in fact, leads to more tractable solutions. This is not the norm, but you should usually start by modeling the biology as well as you can, and then seeing, can I actually improve it by making it, improve the runtime by sort of making some additional assumptions that maybe take me away from the biology. But always start with what would be the perfect solution and then start making simplifications from that. So these are usually the trade-offs. On one side, you have biology and predictability, relevance, correctness, handling all the special cases. On the other hand, you have computability and tractability, with whether there are algorithms that can actually solve it, what kind of assumptions you have to make, how do I even implement this thing, and so on and so forth. Do I have enough memory? And they're typically going to be pulling in opposite directions. There's some examples where this is not the case. Basically, for example, there's a famous chromosomal inversion set of algorithms where if you model the directionality of the DNA segment, you actually get both closer to the biology and closer to tractability. Whereas if you think of these segments as undirectional, it's actually NP complete. So let's now go down that path. Let's basically start with very simple algorithms and then make them progressively more complex to model more and more of the biology without sacrificing computability, okay? So our first formulation is going to be longest common substring. So basically, given two possibly related strings, S1 and S2, what is the longest common substring? That's the longest set of contiguous characters without any gaps. So how long is the longest common substring? Let me hear some suggestions. Who says 1? Who says 2? Who says 3? Good, very good. Who says 4? Very good. Who says 5? It's a great joke about that where Toto comes back and he basically tells his dad, you know, dad's like, what did you learn about today? And Toto's like, oh, we learned about multiplication. And dad's like, okay, what's 4 times 5? Toto's like, 20. And dad's like, great, here's a $20 bill. And that's where Toto says, oh, I should have said 50. So for those who said 5, a little too ambitious. The answer is actually not 3. Congratulations to all those who found 3. TCA, for example, is the longest common substring of length 3. But some of you found 4, right? What's 4? Toto could have earned more cash today. All right, so what did I do? What's the algorithm? Can somebody raise their hand and say, how did they solve it? Yeah. Yeah, slide them past each other. What's the runtime? I don't know, I use my hands once every now and then. So what's the runtime of sliding them past each other? What are the operations? So you basically choose an offset, that's sliding them past each other. And what do I do at every offset? I compare every character, right? So basically, the algorithm is how many offsets are there? N plus M, so N plus M offsets. And then how many things do I check at every offset? N, okay? So it's an N squared algorithm. Everybody with me? So basically, what we're solving is a problem that is not super biologically useful because we don't allow gaps, and gaps do happen in evolution. And then our runtime is N squared, okay? Can we do better? Well, let's look for the longest common subsequence. Basically, given two possibly related strings in S1 and S2, what is the longest common subsequence, not substring, and a sequence allows gaps, okay? In this particular case, I can actually include 1, 2, 3, 4, 5, 6 characters, okay? So the longest common subsequence can basically take the A from here and the G and T from here and then include a gap and then take the TCA from here, okay? So who can give me an algorithm that can solve the longest common subsequence? Raise your hands. Not all at the same time. Yeah, go for it.
So for each base in sequence 1, you take that base, compare it to the other sequence 2, and once you find a match, you see how long you can go continuing to match, and then you have that as like your optimum, and then you continue until you find like the best.
That's great. That's great. So for every position, I basically search all up with the other positions. I find a match that gives me order n, basically, for every starting position. And then what do? I see how much longer I can make it, basically contiguously. And then I may have to choose to open a gap or not at each position. So I have to make a binary choice at each position. So I have 2 to the n binary choices, right? And at every binary choice, I have to search n positions. And maybe on the other side, I have to also choose to open or close a gap. which basically gives me another 2 to the m choices, right? So it's 2 to the n plus m. Okay, can we do better than that? So that's exponential. That's basically the very definition of an exponential algorithm. Basically, if I have 2 sequences of length 1,000,000, it'll take me to 1,000,000 to align. And how many atoms are there in the universe? About 2 to the 78. So it'll need a lot of computation for 1,000,000 long sequences. And the human genome is actually 3 billion bases. 3 billion bases. So a lot more than that. So this is the exponential problem that we're going to have to solve today. And we're going to solve it in polynomial time, right? Who thinks this is kind of cool? Yay, awesome. So this is basically the longest common subsequence. You basically have insertions, deletions, and mutations. This is exactly the evolutionary operations that we want to model, okay? So this is related to edit distance. We basically have the number of changes needed to transform S1 into S2 using a uniform scoring function. In fact, we're going to use a slightly more complex formulation where our scoring function is not going to be uniform. We're actually going to look at gaps with fixed penalty, insertion and deletion operations, a unit cost for every character that's inserted or deleted, and then we're going to vary the penalties for the edit operations. So for example, if I change an A into a G, because they're actually very similar to each other, I'm going to pay less of a penalty. If I change a T into a C, because they're very similar to each other, I'm going to pay less of a penalty. These are called transitions. When I go between a purine and a purine, or a pyrimidine and a pyrimidine, then these are much more frequently confused by evolution and much more common. So we're going to penalize them less. And we're going to see on Thursday, in 2 days, how this is actually a probabilistic formulation. Basically, penalize them less basically means that the log probability of that event occurring is actually, the minus log probability is lower. And that's related to how common are these events. So there's a direct relationship between the frequency with which things occur empirically and the probability that we assign to them and the log score that we assign to that probability. So all of these things, we will eventually, in the next lecture, interpret them probabilistically, okay? So whereas transversions are things that change a purine into a pyrimidine and vice versa, and these are much less common, and these are much more penalized. Everybody with me so far? So basically, polymerase confuses A with G and C with T much more often, so they're going to cost less. So transitions are going to have lower penalty, and transversions are going to have higher penalty. So the next formulation is basically, I can actually recognize that starting a gap costs a lot more because DNA gets broken up, and then extending a gap of length 31 versus 32 is more or less the same cost, because once it's open, you may include a larger number of nucleotides without as much effort. And once you're done and you've repaired that gap, you then don't insert any more nucleotides. So basically, the linear gap penalty doesn't capture that aspect of the biology, but the affine gap penalty does. So you basically have some big initial cost for starting or ending a gap, and some small incremental cost for every additional character that you add to that gap. You could also have a general gap penalty function, but that's no longer computable in the same runtime. And you could also have frame-aware gap penalty, where multiples of 3 that disrupt coding regions much less are penalized less within coding regions, and so on and so forth. You could also seek duplicated regions, rearrangements, and we're going to learn about that on Thursday. So speaking of this exponential runtime, so basically going back to your algorithm, at every position, I have to make a choice as to whether I include or not a gap. And that's basically a binary choice every time. And if you look at the longest, what I call non-boring alignment, basically alignments that don't align a gap to a gap, because if I align a gap to a gap, I can make it extremely long, but I can also shrink it and basically say if I don't allow gap to gap alignment, then at most I have n plus m positions. And at every position, I can choose to include a letter from one sequence or the other sequence, and so on and so forth. So the longest non-boring alignment has n plus m entries. Otherwise, a gap would be aligned to a gap, and we can just condense it. And this is equivalent to gap placement. So alignment is nothing more than choosing where to put your gaps. Okay. It's another joke. There's this engineer who was brought in to solve this massive reactor that had stopped functioning. This company was losing, $10,000 every minute that this machine was not running, and they needed someone immediately. So they call this MIT professor, who basically shows up, looks around for a while, and then puts a big X on the machine at like some very specific spot, and then leaves. And then they open up the X, and they realize there was something wrong behind there, and they fix it. And then he says, "Could you please send us the bill?" And he sent them the bill for, $100,000. Like, "Oh, can you please itemize it?" And he itemized it, placing the X, $1, knowing where to put the X, $99,000. So, $99,999. So, yeah, basically knowing where to put the gaps is extremely important. It's not just placing gaps, knowing where to put them. So n plus m choose n, ways of choosing the gap one placements. At each position, there's a yes, no answer. There's an exponential number of possible placements, and there's an exponential number of sequence alignments. So you can enumerate and score each of them, but that's way too long. We would need way more spots than atoms in the universe. So we're not going to do that. So we need a faster solution for finding the best alignment. So what we're going to do is use a polynomial algorithm to find the best alignment among an exponential number of possible alignments. Okay? And every time you hear this, it should be a trigger for you guys. Every time I say, "Ooh, how do we find in polynomial time a solution among an exponential number of options?" You should immediately say, dynamic programming. Good job. All right, so we're now going to introduce the principles of dynamic programming using a very simple example. Okay? So what we talked about is comparative genomics and molecular evolution. How do we transform and transfer biological problems into the computational realm? How do we formulate them? And then the trade-offs between them. And then also why alignment is hard even after we've transferred it, because there's an exponential number of alignments. So now we're going to introduce the principles of dynamic programming using a very simple example of Fibonacci numbers. Okay? So top-down versus bottom-up computation, learning how and why there are so many repeated subproblems and how we can order the computation to get rid of them, and using a table lookup. And then we're going to look at a basic dynamic programming recipe of choosing a parameterization, setting up the subproblem space and the dimensionality, choosing a traversal order, building a recursion formula, and then ultimately building your traceback. And then we're going to apply all that to dynamic programming. Okay? So how do we turn exponentials into polynomials? So again, you're here because you love nature and you want to make it better. So this does not get much better than the Nautilus shell. If you basically look at this spiral, it has such an amazingly beautiful form. But even more beautiful than the form is the simplicity of the equation that relates that form. So every year, the Nautilus grows by the same amount that it grew in the last 2 previous years. So it gets better at growing as it grows. And that leads to this formula that basically starts with a square of length of side 1, and then another square of side 1. And then the next square is going to be the sum of those two. And the next square is going to be the sum of those two, and the sum of those two, and so forth. Okay? So that leads to the series of 1, 1, 2, 3, 5, 8, 13, 21, 34, 55, which basically results in a Nautilus shell. Okay? So Fibonacci numbers explain exactly that progression by expressing the 6th Fibonacci number as simply the sum of the 5th and the 4th Fibonacci number. Okay? Everybody with me so far? So they were first proposed by Leonardo Pisano Fibonacci, who obviously lived in Pisa in the 12th century. And at the time, he basically developed this model of rabbits that would, you would first start with 2 little rabbits, they would grow to maturity, and then they would have, 2 rabbits at the next generation. And then the siblings would mate, and they would have more rabbits in the next generation. And then those siblings would grow to maturity, and then brother and sister would have nice little rabbits, and so on and so forth. These are rabbits. These are not people. Don't do this. This is also Rome in the 12th century and Pisa. So anyway, still rabbits. But these things is pervasive. It's not just this, contrived model for rabbit generations. If you look at leaves, they again sort of grow enough, and then eventually they start dividing at every generation. And those that have reached maturity divide at every generation. and then the other ones take 2 generations to reach maturity, and then they divide at every generation thereafter. And then if you look at the Romanesque spirals, if you look at the nodular size, the coneflower spirals, there's actually 2 different Fibonacci series in different directions. The ordering of the leaves as it goes around the plant. Look into your garden, you will find Fibonacci numbers everywhere. So your first program, define Fibonacci. If n equals 1 or n equals 2, I return 1, super easy. Otherwise, I return Fibonacci of n minus 1 plus Fibonacci of n minus 2. Sounds good? Super easy to code. It'll bring your computer to your knees. Why? Because if I want to compute the 1000th Fibonacci number, it's going to be 2 to the 1000. Different processes spawning off before I get a solution. My computer will guarantee to crash, basically. Why is that? Because every single time I want to compute this, I spawn 2 processes. And then every time I want to compute that, I spawn 2 processes. So by the time I get to the 1000th row, I have 2 to the 1000 different things that I'm computing simultaneously. But if you notice, what am I computing? I'm computing Fibonacci of 2 over and over and over and over and over and over and over again, 2 to the 1000 times. This is ridiculous, obviously. And you guys are immediately seeing how you can do better. Who can see how you can do better? Raise your hand. Awesome. Almost as many people as have right arms. So how can I do better? If I'm asked to compute Fibonacci of n, I'm going to say, well, I'm not just going to give you Fibonacci of n, I'm going to give you all of the Fibonacci numbers all the way to n. And that's going to cost me much, much less than simply computing Fibonacci of n by filling out a table systematically of Fibonacci of 1, Fibonacci of 2, and then from Fibonacci all the way to n plus 1, because that Python is weird and ends there, I'm going to fill out the table by simply summing up the last 2 numbers. Okay? Notice that right now, I'm just computing 0, 1, and then 2, 3, 4, 5, 6, exactly once each. Okay? I'm basically building a table and then using that table because all of the smaller subproblems have already been computed as soon as I need a bigger problem. Okay? Raise your hands if you're with me. Awesome, great. Do I need the full table? No, I only need the last 2 entries, right? So basically what I can do is simply create 2 entries or 3, I don't know, some generous, and then, you know, fill up the 3rd one and then throw away the other 2 and so on and so forth. Okay, so I can just cycle around. Sounds good? Okay, so what did we do? We basically decided to reveal the identical subproblems, to order the computation in order to enable the reuse of our results, to systematically fill in the table of the results from the bottom up, and to express larger problems from their subproblems. And the ordering of the computation matters greatly. The naive top-down approach was very slow. The results of the smaller problems were simply not available. So I ended up repeatedly computing the same thing over and over again. And then the systematic bottom-up approach was very successful because I filled in the table of the subproblem results in order, and I looked up the solutions instead of recomputing. Okay? Who feels that they've learned something so far? Raise your hands. Okay, awesome. Who feels that they knew it all already? Okay. So everyone who has a right arm learned something. That's great. So dynamic programming is much more complex than what I just showed you. Okay? Dynamic programming is usually an optimization problem. It doesn't just compute things. It makes optimal choices. And we're going to see that in the context of sequence alignment. It's applicable when there is an optimal substructure, when the optimal solution to a problem instance contains within it optimal solutions to the subproblems. And it is very important when there are overlapping subproblems. In this particular case, there was an exponential number of reuse of all these subproblems, but there was a limited number of distinct subproblems that were simply repeated many, many times. And that's when dynamic programming really, grows. So as I mentioned, it's for optimization problems. Fibonacci was not an optimization example, where the optimal choice is initially made locally by choosing maximum pointers, and then the score is added through the search space, and at the end, you trace back through these local maxima to find the globally maximum solution. And we're going to see this in much more detail. And it's also middle of the road in range of difficulties. There are some problems that are much easier, where a greedy choice is possible at each step, and we just go greedily choosing the whole way, never do any traceback. Dynamic programming requires a traceback to find that optimal path. And then there are much harder problems where there is no optimal substructure. For example, a subproblem may be dependent on other subproblems, and then you cannot solve that anymore. Okay? So dynamic programming basically has optimal subtraction overlapping problems, but greedy algorithms allow a greedy choice property where I can locally make a choice each time. Whereas here, I make I have to remember a lot of local choices to then reconstruct the globally optimal choice. All right, so dynamic programming in practice requires 5 different steps. The first is to find the matrix parameterization. What is the number of dimensions, the number of variables, which variables are there? So in the Fibonacci example, it was 1-dimensional, and the variable was simply, which Fibonacci number am I computing? In the sequence alignment problem, it's going to be 2-dimensional, and the variables are going to be which length prefix am I computing? Make sure that the subproblem space is finite, not exponential. I've seen this before where people are like, oh, I'm just going to systematically compute every subproblem. And there's an exponential number of subproblems. You'll never be done. With a big enough size problem, you will just never be done. In that case, memoization is one approach. If not all the subproblems are used, but some of them are going to be reused, but you don't know which ones, you can simply hash the parameters that you're throwing to the routine. And then if it sees these parameters again, it's going to remember the solution. And maybe if it's still an exponential problem, but you don't have to explore the full space, it might still work. So if your use is not extensive, perhaps dynamic programming is not the right solution, because here you're going to have to compute every single subproblem. You're not going to have the choice to sort of compute a subset. The traversal order is something that matters a lot. So for Fibonacci, we only computed the next number after all the previous numbers were already computed. And because it was 1-dimensional, we could use that with growing size numbers. This is bottom up, but it's not always obvious. So basically, when you design your own dynamic programming problems, you're going to have to think about how do I explore that space to make it work. And then the last step, or the most important step perhaps, is the recursion formula, which is how do you actually compute the larger problems from the subparts? What is this F? And that is, sometimes going to be very difficult. Okay, so computing this F function. And then the last thing is remembering the choices. So this F function is going to include some minimum or maximum, and you're going to have to remember the pointers. You're going to need some representation for storing these pointers if this is, a complex polynomial solution. And then when you have all that, you start computing. You basically systematically fill in the table of results, find your optimal score, and then trace back from that optimal score to find the optimal solution. Who's with me so far? Great. All right, so we've talked about sequence alignment. We've talked about the principles of dynamic programming. And specifically, we saw a very simple example. That's not even an optimization problem of computing Fibonacci numbers, top down or bottom up. We saw the huge number of repeated subproblems and how to order the computation and how to use table lookup to not have to recompute. And then we saw this very simple recipe of the 5 things that you're going to have to do every time you're faced with a dynamic programming problem. Namely, choose your parameterization, choose how to, define the subproblem space, choose how to explore that subproblem space, and then define your recursion formula and complete your traceback. Okay? Now is a mandatory stretch break. So I want everybody to stand up and stretch for 1 minute. All right, reach up to the sky. Everyone, go left. Look at your right neighbor from the left. Now look at your left neighbor from the right. Yes, that is possible. Stretch your arms, stretch your neck, shoulders. You think I'm doing this because I'm nice. I just want more blood to your brain. I'm pushing calories into that massive compute power of yours. All right, hop a little bit. Great. All right. So meet at least one person. Say hi to your neighbor. Learn their name. Oh no. I'll send you the recording as always. Yeah. All right. This part is hard, so I want your brains to be fresh. I hope 60 seconds is enough. But now we're going to combine these relatively easy solutions, easy topics, basically on one hand, sequence alignment, and all these complexities that we learned there. And on the other hand, dynamic programming, of which we saw a very simple example, and we're going to put them together. You guys ready? How do we apply dynamic programming to sequence alignment? The idea is the following. If I want to compute the score for the alignment between A, C, G, T, C, and T, A, G, I'm going to do that as a function of the score for smaller alignments, okay? Don't worry if you don't get everything I say the first time around. I'm just going to repeat myself for the next 20 minutes, okay? I'm just going to say the same thing over and over and over again in a bunch of different ways, okay? So what we're going to do is realize that the score is actually additive. That basically every single time I'm computing this score, extending it is simply some addition to the previous subproblem score, okay? So what I'm going to do is I'm going to compute my best alignment recursively. The best alignment of going through this CG nucleotide pair, having them aligned to each other, is going to be the best alignment all the way to here and all the way to there, plus whatever score that is, and the best alignment all the way to there and all the way to there, okay? One way to think about these ij parameters is a matrix where I'm looking at increasing length prefixes of the first sequence and increasing length prefixes of the second sequence. So I have S1 all the way to I, and I'm aligning that to S2 all the way to J. And that score I will save in the IJ entry, okay? And then in order to compute the best score of the whole sequence S1 to the whole sequence S2, I will have to compute and that score that goes through C and G, that basically aligns the C nucleotide to the G nucleotide, that score better be made up of the best way of aligning this and the best way of aligning that, okay? Why is that? Because if this was not the best way, then I could find a better way that gives me a better score. And then I could just put that together with whatever I have here and get a better score, leading to a contradiction, okay? Basically, if that's the case, then I can get a better score overall, and therefore this could not have been the best score. So the best score that goes through this point better be the best score all the way to there and the best score all the way to there. Who's with me so far? Great. So basically that's this optimality constraint that basically says that optimal solutions are made up of other optimal solutions. But it's a lot more complicated than that. Why? Because that optimal solution does not necessarily need to go through this position. It may go through some other position, okay? So we're gonna have to calculate this for all possible midpoints, okay? So therefore, if I were to write a top-down program, the same way that I did for Fibonacci, I would need to compute an exponential number of subproblems. But if you look at this 2D parameterization, there's only a finite number of subproblems. It's all the ij points that I'm crossing through, okay? So basically, for every alignment, I have to go through some pair of nucleotides that are aligned, and there's only n times m pairs. Who's with me so far? Great. So the key idea is that in order to calculate this ACGTC versus TAG alignment score, not just some score, but the best possible score that I could obtain to align those. And I'm not gonna worry how I got that score. I'm just gonna worry about what that score is. To calculate that score, the maximum alignment score, I'm gonna choose the maximum among 3 options, each of which builds on a smaller sub-problem. The first option is that I'm gonna build on the alignment between ACGT and TA. I will assume that I've already, that all shorter alignments are already computed. And now I'm gonna compute a longer alignment. Everybody with me? So then I'm gonna show that I can use those smaller alignments to compute the longer alignment. And by induction, I will therefore be able to compute anything. Because if I have the smaller alignments and I can use those to compute a larger alignment, then I can effectively compute alignments of any length. Okay? So the first option is to extend the ACGT alignment by adding a C to one sequence and adding a G to the other sequence. Okay? So by extending the S1 prefix by another letter and extending the S2 prefix by another letter. Everybody with me here? And what's that score? Well, that's simply whatever score I had here between those two, plus the score of aligning C to G. And if it's a purine, it's one score. If it's a pyrimidine to pyrimidine, it's another score. And if it's a transversion, it's a different score. Okay, so basically, I can just look up my score matrix of what is the cost of aligning each of ACGT to each of ACGT. Everybody with me so far? So that's option number 1, where I'm aligning those 2 nucleotides to each other. What's another option? Yeah.
You take one nucleotide from one sequence and then create a gap with the other one.
Perfect. Okay. So solution number 2 is I'm just going to extend this sequence by 1, and for the other one, I'm just going to put a gap there. Okay. This is still a smaller subproblem. I'm basically now building on this 5 long prefix versus 2 long prefix, and then adding up the score of a gap, the cost of a gap, basically, to whatever maximum cost, maximum score I had for the previous sequence. Okay? Who's with me so far? Nice. And then the last option is I add a gap to the other sequence. Okay? So basically I extend this, ACGT with a C, and then I add a gap here. So I can build up on one of 3 smaller subproblems. Basically this 5-3 subproblem can be computed either based on the 4-2 subproblem, or based on the 5-2 subproblem, or based on the 4-3 subproblem. This is kind of cool, right? I don't have to worry about sequences anymore. I just have to worry about entries in the matrix. And I just have to fill that matrix from the bottom up to make sure that I have all of the smaller subproblems when I need the bigger subproblem. Okay? Are there other options? Could I have constructed it some other way? Do I need to look at more than just 3 options for the smaller subproblem? Everybody's nodding no. I'm going to ask you, why don't I have to look at the ACG versus TA subproblem?
You already looked at it.
Beautiful answer. I've already looked at it. When? Exactly. When I computed this, I already built on that answer. So whatever way I could use that has already been incorporated into here. Okay? Everybody with me? This is kind of cool, right? We're basically now starting to move from this totally amorphous exponential space of possible, gap placements to just entries in my matrix. And the rest is going to be, well, the details of how do I put all that together. So option number 3 is extend this by adding a gap. Option number 2 is extend the other one by adding a gap. Option number 3 is extend both by adding a sequence and then compute the match mismatch score. Okay. So we're going to store all of these scores in a matrix parameterized by which prefix I'm looking at and which prefix I'm looking at. And we're going to fill the matrix systematically from the top left to the bottom right. And when I get to the bottom right, this will be the score of the best possible alignment of the nth prefix of S1 and the mth prefix of S2, which is basically the whole sequence S1 and S2. And that will be the score of my optimal solution. Not quite my optimal solution. I will have to then trace back to build my optimal solution. Okay, so these are the 4 entries that I just looked at. How to compute 5.3 using 5.2, 4.2, and 4.3. This basically goes in here. My S1 sequence goes there. My S2 sequence goes there. The ith prefix is indexed by I here. The jth prefix indexed by J here. And I'm computing 5.3 using 4.2, 5.2, and 4.3. Cool. Now, any path through this matrix effectively tells me a series of operations. Every time I traverse this matrix diagonally, it tells me that C was aligned with C and T was aligned with T and C was aligned with G. These are the diagonal paths through the matrix when I'm adding nucleotides on both sides. All the vertical paths are basically a gap in this sequence, but a character in that sequence. Every time I go straight down, I'm adding a character here, but I'm not adding a character there. And every time I go straight to the right, I'm adding a character in this sequence, but I'm not adding a character in that sequence. Okay? So I can basically look at any path through this matrix and paint an alignment between 2 sequences. So I go down, I add a T with a gap, I align the A to the A, I go this way, I add a C, but no other character in the other sequence. And then I go this way, I add a G and a T and a mismatch between G and C. That's a mutation. And then I add a gap in one sequence and an A in the other sequence. And then TCA. Okay? So we basically have every entry in this MIJ matrix corresponding to the best alignment score between the ith prefix of S1 and the jth prefix of S2. And the best alignment is simply the best path through the matrix that gives me that maximum score that I get at the bottom right. Okay? Yeah? It's cool, right? It's fun. Like, you know, here's another duality. There's this duality between paths through the matrix and alignments through S1 and S2. So there's this computational structure that we've just built of this matrix and this biological interpretation that we're building of scores and alignments. So I went on Wikipedia this morning and I found a cool animation and I copied the GIF and I put it in there. So animation credit Wikipedia. What is it doing? It's basically, you know, filling out exactly that score. It's basically going from that initial matrix down at every row, adding one more series of scores. And how are these scores computed? They're computed based on +3 match and a -3 mismatch, and you know, a gap score of 2. Okay, so every time I'm here, I can basically say, well, how did I compute this? It was the best choice of adding a gap here, or adding a match mismatch there, or adding a gap here. Sounds good? And the maximum was only given by the diagonal entry where G and G are matched to each other. Okay? And then, oh gosh, I wish I could stop these animations. And then same for here, G matches G, so I get a 3, G matches G, so I get a 3, and so on and so forth. Okay? So I go progressively filling out that matrix all the way down to the bottom. And then for global alignment, I have to start at the bottom. For local alignment, I can just start at the maximum character. And then I trace back the pointers. Which of these gave me the best choice here? It wasn't the 8, it wasn't the 8, it was really the 10 + 3 that gave me that choice. So I can basically start reconstructing my best optimal solution by walking back through these pointers. Okay? And then as I construct that solution and get to this 0, that no longer has a pointer, so I have to stop there. Okay? So I initialize the matrix with zeros everywhere. I fill the matrix from the top left to the bottom right. Why? So that every single time I need to compute a number, the top left, and the left entries are already computed. And then as I fill the matrix, every time I choose a maximum of the three, I basically say which number gave me that maximum. Here, the maximum of 4 can be obtained either by adding 3 to the 1 diagonally, or by subtracting 2 from the 6 horizontally. So I can choose both of them. And either of them would give me an optimal solution on the way back. So there are multiple paths that give me this 13, which basically correspond to multiple alignments, okay? And as I go back, every diagonal pointer matches 2 characters together, and every vertical pointer inserts a gap in the top sequence, but a character in the left sequence. Okay? Who feels that they've learned something? Yay, awesome. And who feels that that something is actually cool? All right, so here's the sequence. Here's the resulting alignment. Here's the matrix as I fill it out. And here's the trace back of that matrix, okay? So I'm just gonna now repeat the same thing over and over and over again with increasing levels of detail, okay? So we're basically computing this entry as a function of simply smaller entries. We are using only local update rules. Why? Because otherwise my computation explodes. Here I have an n by m computation. Why? Because there are n by m entries and every single entry is computing only local stuff. So I have a constant number of things to do. If instead I was looking at order n, it would be a cubic algorithm, okay? So the power of using only local update rules is that it's actually, you know, order n squared. Just like Fibonacci numbers, I'm only using some small number of local operations. And the table lookup avoids repeated computation. To compute the score of this cell from the smaller neighbors, I had a maximum choice. That's where the optimality comes in. Remember, I mentioned Fibonacci didn't have any optimality choice, whereas dynamic programming in general has some maximum in there. Here's where the maximum sits. I'm basically making locally a choice and remembering the options that gave me that best choice. This is a local choice, not yet a global choice, because I don't actually know that I'm going to go through this entry. So I can't just say, oh, I got the best alignment here. This entry just might never be part of the best solution. Whereas these entries are part of the best solution. All of those, I computed them, but they're not part of the optimal solution. The path does not go through there, okay? There were only 3 possibilities for extending by one nucleotide: a gap in one species, a gap in the other species, or a match or a mismatch. And the reason for that is that all smaller subproblems I had already incorporated in those 3 subproblems that I had computed. And I computed scores for prefixes of increasing length. We start with prefixes of length 1, we extend by 1 each time until all prefixes have been computed. And when you reach the bottom right, the alignment score of this full prefix and this other full prefix is effectively the alignment score of the full 2 sequences, okay? And then we trace back to construct the optimal path, okay? So we computed all alignment scores from the bottom up. We defined this matrix of prefix alignment. We filled out the table recursively. We expressed the alignment of, you know, this and that together to compute this. There were only 3 possibilities. You know, it took us order 1 time. And then the proof of correctness is this cut and paste argument. So looking back through the 5 bullet points of a dynamic programming, the matrix parameterization, that was basically choosing the score of the prefixes. So it was this IJ parameterization that was basically telling me the length of characters that I include from one sequence and the length of characters of the other sequence. So I could have instead used all substrings. So a 4-way parameterization from I to J in one sequence, from K to L in another sequence. And that would have been a 4-dimensional entry. Occam's razor, let's just use a 2-dimensional matrix. No need to do that. I wanted to make sure that the subproblem space is finite, not exponential, and that's just n squared, quadratic, which is polynomial, not exponential. There's only so many combinations, so many pairs of prefixes. And then the traversal order was anything that gives me those top left and top left entries. So I can go down the columns, I can go down the rows, I can fill it out diagonally, anything that makes sure that those three are already available. The recursion formula, that was the hard part, okay? We need a formula for computing Mij as a function of the previous results, okay? So it's a single increment at a time, and that's this formula here. It's basically the maximum score of the ij alignment is the maximum of this alignment plus whatever score I get for aligning this character to that character, or that score, which is already the maximum for going all the way to there, plus a gap penalty, or that score plus a gap penalty, okay? So that's the recursion formula. It's quite simple. And then the score depends on the pair of characters at positions I and j. Okay, everybody with me with the recursion formula? Yeah.
So can you not implement a fine gap penalty with local update rules?
You can. This is a linear gap penalty, and a fine gap penalty absolutely can be used, but you have to include 3 matrices or 2 matrices. That remembers if I'm already in a gap, then I can just keep going, adding that gap. And if I just close the gap, I will pay a different score for opening a gap. So you need to remember some state. And we're going to talk about state when we get to HMMs next week. So basically the state of this system is 0. It's basically whatever entry I'm in. But an additional state would be which matrix I'm in. So I could have one matrix where I'm already in a gap, another matrix for I'm already in a gap in S1, or I'm already in a gap in S2, or I'm not in a gap. And then if I just open the gap in one sequence, I go here, and I keep traversing there. And if I just open the gap in another sequence, I go there, and I keep traversing there. And I could get away with only 2 matrices because gaps don't ally to gaps. So if I've opened a gap in one sequence, I will never have to open a gap in the other sequence. So I could get away with just 2 matrices. So state of, you know, binary choice of am I in a gap or not. Does that answer your question? Yeah. Any other questions? Okay. So number 4 was basically the recursion formula of computing Mij as a function of smaller results. And the single increment at a time allowed me to only look at left top and left top, corresponding to 3 options. And then I scored each case depending on the gap match, mismatch penalties. And then, most importantly, I remember the choices. So in that function f, I included a min or a max, and I remembered which of the 3 cells gave me such a maximum. Okay? So here's a very simple example again, where I've written all out in terms of, you know, what gives me these optimal solutions. This is superseded by the Wikipedia animation, which is much cooler. But this is not superseded by anything in the world because I made it. So this is computing dynamic programming using Excel spreadsheet. So in full disclosure, I have no idea how to use Excel. I had to look up these functions as I went along. And in also full disclosure, I had no idea this was actually going to work. I just went one matrix at a time. And I was like, I could just compute the score. So here's how this works. You basically type your sequence here, and you type your sequence here. Computing the score of matching this character to that character is super simple. I basically created some cells here that basically tell me what is the score of aligning A to A, G to A, C to A, T to A, etc. Okay, this is exactly the penalties that I mentioned earlier. So I get a -1 if I go a pure improvement transversion, and I get a moderate penalty / lack of penalty for aligning a pure into a purine or a purine into a pyrimidine, and then I get a match reward for matching characters. Okay, and then I have my gap here. So you enter your parameters here, and then you let it loose, and you type your sequences, and this is just the score. Every time you have a match, TT, AA, etc., you basically have 1, and every time you have a mismatch, you have 0, or a transversion, you have 1. Okay, that's just looking at one character at a time. Then comes the actual score matrix. So basically, I could have just written that, but it's simpler to first see this. So then at every position here, what do? I basically choose the score at that position based on whether it's the maximum here, or is the maximum between this plus a gap, or this plus a match, which I get from that matrix, or this plus a gap. Everybody with me so far? So those two matrices are trivial, okay? And here's the corresponding code. So basically, again, I don't know how to use Excel. I had to look it all up. But, you know, this is just the index in that matrix up there. And then, you know, if there's a match, so basically looking up that character and then looking up that character. And then the code here is, I'm a geek, I agree, fine. You can laugh at me. So this is the maximum alignment score. Basically, it's the maximum of these three choices. Sounds good? Super trivial, right? Who's with me so far? So basically first looking up and indexing and then doing that, okay? So basically this basically tells me, you know, find where the A is in that row, find where the T is in that row, in that column, and then finding the index of these two inside that matrix. Super trivial, okay? So those two we're super cool with, okay? Now, can I actually write down the pointers? That's easy. All I need to do is compare the score that I get here with each of these three options. And if that matches this, then I add a vertical. And if that matches the diagonal, I add a diagonal. And if that matches a left, I add a left. Okay? So that's simply concatenate. If this put a vertical, if that put a horizontal, if that put a diagonal. Everybody cool with this matrix? That's the matrix of pointers. All right, now let's trace the maximum path. So when I get to the bottom left, I just follow pointers. So basically, is the ij part of the optimal? Well, we can just look as to, you know, the bottom right. If it was pointing to me, then I'll just be happy and I'll just light up, okay? But I'm not just lighting up here, I'm actually doing the sum. So I'm not just calculating the optimal path, I'm calculating the number of optimal paths that go through that matrix. Is everybody with me here? Okay, and the last part, it's a little crazy. Can I actually calculate the actual alignment? Can I actually do the sequences? And the answer is yes, apparently. So, you know, every time I go to the left, I choose to concatenate the corresponding character from that sequence up here, or I choose to concatenate just a gap with that sequence, and I construct one, and I construct the other, and then I produce the best alignment here. This is kind of cool. So you can actually find the spreadsheet here. I have it loaded up there. So you can actually change your thing, and you can actually compute stuff, and you know, change the letter T, and see everything get recomputed, and so. Right? Fun stuff. All right. So that's genome alignment in Excel spreadsheets, just simple. So that's what's crazy, right? Like, I don't know if you guys saw that they were able to compute 42 as a sum of 3 cubes. Yeah, I'm 42. I organized the Meaning of Life Symposium because 42 is also the answer to life, the universe, and everything. And it's kind of cool that like on my birthday year, it's like we finally have that answer. It's one of those things where it sounded like you needed to go through this crazy exponential number. I tried to check the solution to the 42 thing. Excel cannot do that. Powering 3 numbers with like 12 digits each is not possible for Excel. It truncates the last 2 digits. And that's program is still able to compute dynamic programming. Right? We basically took this exponential search space and suddenly, whoa, it's just an Excel spreadsheet. You just, you know, type them in. So that was computing the optimal solution and then tracing back. So the trace back basically needed to just remember these pointers. And to remember the pointers, you needed to actually store the entire matrix, okay? Whereas for computing the score, I could get away with less, right? How can I compute the score at the bottom right? All I need to remember is 2 columns, because every single time I'm done computing that column, I will not need this one anymore to compute the next one and the next one and the next one, right? Who's with me so far? So that means that I can compute the optimal score in linear time, sorry, linear space. It's still quadratic time because I have to go through the whole thing, But if I have a million long sequence, I don't have to create, you know, a trillion long or big matrix. I can just do it in just 1 million space. Okay. So let's look at some of these advanced topics. Okay. So if time permits, extensions to the basic DP solution. So the first extension is bounded dynamic programming. Okay. We can now start thinking about solutions in this matrix space. We don't have to look at alignments anymore. And basically, if I have a pretty good score going down this diagonal, I will never have to veer off diagonal and come back. Okay? Because veering off diagonal means basically paying for gap, 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 and then gap, 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 gap. Okay? And there's just simply no combination of these mismatches that will give me a worse score than all of these crazy gaps. So bounded dynamic programming basically says, well, for sequences that are pretty similar to each other, I will never have to veer off course. And therefore, instead of computing the full n by m space, I can just compute the n plus m diagonal times some width, basically this kn space. Who's with me on this one? Raise your hands. Awesome. So this basically says, just initialize it everywhere, and as long as you veer off course, just don't worry about it. Don't go there. Okay? Don't compute anything. So what about linear space? We already saw how, I mean, I just mentioned how we can do linear space by just keeping 2 columns at a time. But the problem is that I need all of the pointers in order to be able to come back. Because then all I will have is the score. I won't have the pointers. Can I do linear space alignment with tracing back of the scores? And that's where it gets a little geeky again. How do I remember the pointers? There's a trick, and if you've understood the lecture so far and you're sort of feeling comfortable, remember this feeling right now. Just memorize that feeling. It's a great feeling. Your brain just might explode. Be careful. All I need to do is compute the pointers from the midpoint that gave me whatever score I have here and how I get back just one way. And the pointers, I can just compute the exact same dynamic programming solution in this matrix here, going from the right to the left, and just remember the score for the right half of that sequence and the pointer given to that. And then I can add this column to that column. That basically gives me the maximum score for the middle column, which basically means the maximum score going through this character of that sequence. And whichever character has the maximum here is the character that I'm going to have to go through. So I just remember which character I'm going to have to go through and which way I go, up, left, or to the right, and so on and so forth. Everybody with me so far? So I can compute the maximum midpoint. And that's where it gets really awesomely cool. I can then cut the matrix, never bother computing here, never bother computing there, because I know where I went through. Okay? And after I've done that, I just recurse here, find the midpoint, figure out how to recurse, and then cut the matrix further and further and further. Okay? This is 1 plus a half, plus a quarter, plus an eighth, plus a 16th. It's that turtle that never gets to its goal. I never get to 2. Okay? So that is basically only a factor of 2 worse in computation time, but a factor of n better in space. Okay? So instead of computing the full n by m matrix, I just compute 1 row at a time, find the midpoint, break it up, find the midpoint, break it up, and so on and so forth. Who think this is kind of cool? Who got this, by the way? Raise your hands if you got it. Who was super, super comfortable right before and remembers that feeling greatly? Awesome, great. Remember that feeling. What did we learn about today? We learned about dynamic programming, about reuse of computation, about how to apply dynamic programming to sequence alignment, and then some really, really cool ways of doing this in Excel, in linear time, and in bounded space. Okay? Good stuff? See you guys on Thursday. We're going to learn a lot more stuff.
So